Oh, looks good.
Okay, so like I said, so CWAC 2 has been going for 14 years now. We're growing at about 1.5 million to 1.8 million records per year, which is a great level. And at the moment, the database represents about 10,000 hours short of 1 million hours effort from the observers to collect this data. Okay. CWAC has been running since 1992, when the South African government approached us to start the project and to monitor the wetlands. We have about 347,000 records, monitoring 700 sites, or just over 700 sites per year. Our longest data set actually goes back beyond 92 for Longabon wetland, which is about 40 years, the data set. We count twice a year. It's all citizen science-based, so it's all volunteers counting. We collect, obviously, the count data for each species, but we also do collect a bit of data of the environment as well. But that is, we need to expand on that quite a lot. Yeah. Our infrastructure systems at UCT, we host all of our own servers. We are not cloud-based. All our machines are Supermicro servers. They are very well equipped with Intel Xeon processors and enough RAM to do everything we need. They are all backed up daily, and those backups are put into the cloud for safety. Okay. Incoming data we get data primarily through the websites and through a mobile app, BirdLasser. BirdLasser is a third-party app that was designed around the bird atlas, but has been extended now, and it's currently been extended to collect CWAC data as well. And it supports a whole bunch of other causes, which is quite nice. Data comes in through our primary API server and web server. Both are firewall protected. We have multiple firewalls on the system to protect our data server. Yeah. At the bottom, I don't have a pointer on this thing. Can you believe it? There. At the bottom, that we have a new server that we just put in, the Pipeline API server. This is now specific for this BIRDIE project, where we are designing API calls to meet the needs of the seeks analysis and stuff like that. Okay. Then, right. Data coming into the project goes through quite an extensive validations process. This helps us ensure that we don't get, or we're not serving out of range records, or we're not serving data that hasn't been checked. We have, how many is this? 1, 2, 8 levels of data checking to come in. So every record comes in. The data is compared against the data dictionary to ensure that it's a valid species, a valid site code, the date's all correct, things like that. That's just basic to ensure that we're not getting any SQL injection data. It's compared to the CWAC 2 database first, and that validates the range of the species to ensure that it is in range. Goes through various levels of spatial checks to ensure that we are actually registering the correct species in the correct place. If it fails all of those systems, we deem it to be an out-of-range record, then it goes into the out-of-range system, which on this slide is the ORF system. This query is then sent back to the observer, where they have to validate and justify their species by giving descriptions of its location, habitat descriptions. And those are then validated by a regional expert who interacts with the observer and then makes a decision there. That the regional expert will make a decision whether it's a valid record or to reject the record. Valid records are then put into the primary database for use. Rejected records are binned. Okay. The database then feeds those records into our primary API system, which then that is used by all of our websites and all of our current data products. And then it also will now feed into the pipeline API system, which will then feed the data into the BIRDIE project. And this allows us to serve two types of product. We can serve a raw data product, which goes to other researchers and to anybody who needs it, who can look at the individual raw data. And then we can also then serve the analysis product, which will then come from the data mart. Okay. We've got very simple data access tools built through the API. And this gives you access to the data in a form that is used, can be used very quickly. We serve it in CSV files, JSON, so that'll be useful everywhere. JSON, if you want to use it in scripting languages, and then GeoJSON, which is a GIS-enabled version of JSON. API calls are very easy. We use just a routing system where you designate your format and what data you're looking for. This is CSV and JSON. And then GeoJSON, you can pull straight into ArcView, into QGIS, or any of the GIS packages. They all support GeoJSON now. Okay. API options at the moment, we use two different methods. We use GET, which is then just a straight routed URL route pull on the data. Our default format is JSON, but then you can add a switch or variable onto that to get it in the other formats. We have filtering options on the URLs, and you can filter up to 3 levels deep to reduce your, just refine your searches. We do have a post option, which allows you to stipulate various other requirements. An example here, I've got the content, which allows you to reduce or filter the columns that you're receiving from the data set. So you don't have to get the entire 30, 40 column data set. You can actually only pull out the 10 that you need. These are passed in SQL compliant format and allow you to use aliases to suit your code. So you can rename columns as you're pulling them, which makes it very easy to use. Okay. We do have a list of banned words, so you can't use certain keywords, and all requests are validated against a data dictionary to ensure that the field names are correct and valid. Okay. Example calls at the moment, we can pull out the dictionary, which has got the species list, all our coding descriptions, things like that. Metadata, which describes the data set, as everyone knows. And then you can pull out records. You can pull out the species lists, lists for locations, or you can pull out just the full-on records. All records are, like I said, available in 3 formats and can easily be mapped into any of the scripting or statistical languages. SABIP 2 has the same options available. I am busy normalizing the naming structures between the two, and we will be moving the SABIP 2 API calls onto the pipeline as well, so that it can all be pulled into one system using the same nomenclature. Okay, that's pretty much an overview of the system.
Great. Thanks, Michael. That's fantastic. I don't see any hands up, so I think let's move into the final section for this morning. And the group from the UCT Center for Statistics and Ecology, Francisco, Reyes, and Vernon. I'm not sure in which order, but yeah, thank you.
Yeah, maybe I will start. Hi, everyone. My name is Pachi Cervantes, and just to clarify, Pachi is short for Francisco. And I'm a postdoc at the Center for Statistics in Ecology, Environment and Conservation at UCT, and also at SAMBI. And let me, well, I have to share my screen. Okay, can everybody see that?
Yes, we can see that. Thanks.
Perfect. Then I need this full screen mode. Okay, so Rez and I don't know if Bernard's going to jump in at some point, are going to be talking a little bit about the statistical models and statistical routines that we're going to be implementing in the BIRDIE project. And yeah, this is going to be happening at SIG mostly, but we're working closely together with the Fitzpatrick Institute and so Pike University and also with Francis and yeah, the other parts of the team. So basically, this is the structure that we've already seen. Francis showed us earlier, where we've got the CWAC and CWAC data coming into the pipeline. And then we've got some cleaning and validation of this data. And then we take that data and we implement some statistical routines that are supposed to run periodically as new data comes in. And out of that analysis, we are hoping to get some data that it's in a format that it's easier for interpretation and that contains perhaps some extra insights. And then that is perhaps what Francis was referring to earlier. So we get some raw data that's available from the Fitzpatrick APIs, from the ADU APIs. But then we will process that data and we'll generate new information that will be available in the data mart. And then that information will in turn be available for web apps or user interfaces that can be used for the final decision making. Types of decisions that need to be supported. Very quickly, Nancy touched on this. So there are different reports and assessments that these pipelines should be useful for, and they are at different levels. So from site reports and management plans, all the way to provincial and national reports, red listing, national biodiversity assessments, and international agreements. There's also possibly the need for research and other type of questions. So in order to define our statistical routines, we need to actually define what we want to look at. And at the moment, we have defined a few metrics that are the metrics in a sense: distribution of species, the abundance, so population sizes, how populations change over time, and then also species diversity. The interesting part is that we also need to integrate these metrics for different populations, and in the sense that we need to make this scalable. So we want to understand the population size at a certain site at the local level, but we also want to try and incorporate or integrate different sites to get a sense of what's happening at the provincial level or the national level, and also integrate different species, like, for example, to understand what's happening with different guilds, like or how different trades relates to each other. And then we need to understand that we're dealing with citizen science data, which is very powerful because we've got lots of people out there collecting data and covering very large extensions. but on the other hand, these observations are prone to have some errors. So our observers are not going to count the exact number of birds, for example, or might miss a species that is present somewhere. So we need to deal with this in the statistical analysis. So yeah, the main perhaps type of model that we're going to be using perhaps falls under the category of a state space model. That is a model that describes how processes change over time. So it's dynamic models and account for observation error. It's like a very general model definition. And so in this diagram here, for example, we would see that we have some process in the square boxes that evolves over time. And so we see that this mu, which is the process value, could be, for example, population size. At time t depends on the population that we had at time t minus 1, and the population at time t plus 1 depends on the population at time t, and so on. So in other words, the process that we're interested on evolves over time, and it is correlated over time. It changes with a certain rate. That can also change over time. But then we usually don't get to observe exactly what we wanted. So we're not able to count all the birds, for example, in a population. So we count certain amount of birds, which is represented in the circle up here, and the amount of birds that we count is obviously, or hopefully, related to what the population size is, but it's not exactly the same. And so we've got certain observation error. So our interest really is not on the counts, it's on the process. And so we need certain statistical methods to try and filter out these errors to some sort, or at least try to describe it. And the key thing here is that you see that there's no arrows going between the observations. So this means that the observations are not, or so the errors are not necessarily correlated over time. They are correlated because they have something to do with the process, which is correlated. But other than that, they don't depend on previous stuff. And that is the information that the model uses actually to try and filter out these errors. So this is all a little bit abstract, and I wanted to show a more specific example. So when I refer to a process, I refer, for example, at the population size. So this is a hypothetical population of species that is described by this blue line. And it changes over time continuously, right? But we don't get to observe that all the time. We only get to observe this in this particular example at 5 points in time, which are represented by the black dots. And the dashed lines over here represent the discrepancy between the actual population and what our observers actually record. And as we can see, for example, from time 1 to time 2, the population actually increased by quite a bit. But if we just look at the raw counts, we wouldn't see that increase at all. And for example, we would see a really big change between time 3 and time 4, but it doesn't exactly correspond to the profile of the actual abundance. So these might not seem like a big problem, but if, for example, we want to relate the counts to a temperature profile, and the temperature profile more or less follows the blue line, then we wouldn't be able to attribute perhaps with the confidence that it would deserve the change in the population. For example. So we want to try and estimate what the actual population is, and not just use the raw counts. And so that's basically what we're going to do for the CWAC data. And then I'm going to explain that a little bit, and then Rez is going to talk a little bit about the occupancy models, which don't deal with abundance or population sizes. They deal more with whether the species is present or not at a certain site, and therefore useful for defining distributions. So for the CWAC data, Michael covered this more or less, but it's basically a project that has 2 counts per year at least. So we've got a summer count and we've got a winter count, but then there are some other additional counts. Because these additional counts are not all the same at different sites, we are only using summer and winter at the moment, but we are thinking of how to maybe incorporate those additional counts somehow. Then, yeah, when a CWAC site is monitored, then it must be well defined in terms of its spatial limits, and the procedures that the observers use must be consistent across time, so that we can actually compare across years. Okay, this is a bit more complicated. It is a similar diagram as the one we saw before. It's just a little bit more complicated because we don't have we don't have one state process, we've got 2. So in the previous slide, we only had 1, but here we have the white process here, which is the summer, the population in the summer. And then we have this for the black process, which is the population. The population size, let's say, in the summer will provide us with some summer counts. And the population size in the winter will provide us with some winter counts. Other than that, is the same principle that we observed in the previous slide. So the process of time t depends on the process at time t minus 1. The process at time t plus 1 depends on the process at time t. So because we've got movement of birds, and we've got Palearctic migrants coming in the summer, and we've got some inter-African movements as well in the wetlands. We need to somehow obtain the winter populations from the summer populations. So we need to distinguish somehow that in the winter the population is different from the summer. And this is where this lambda comes in. So this lambda is kind of like the proportion of birds that we would expect that would stay during the winter months, the proportion of the summer population that would stay for the winter. And that proportion is also correlated over time, and these betas, by the way, are also correlated. This is pretty much the same model that was used by BIRDIE et al. In 2017 in this paper. And just to show you some example, I thought this was a nice example of what the output that we're getting looks like more or less at the moment. So on the top panels here, we've got the winter, we've got the summer population, well, the summer counts, let's say, it's the green dots, and then we've got the winter counts, which are these purple dots over here. And this corresponds to the glossy ibis population at Barberstein, which is our current pilot study site. And we've got the solid line represents our best estimate for the population size in the log scale. Don't worry about that. So, and then the dashed lines represent the uncertainty that we have around that estimate. And we have uncertainty for 2 reasons. Firstly, because we're modeling this line, and secondly, because we've got observation error. And I thought this was interesting because this line is wiggly, as you can see, and we could actually just draw a straight line through the data here. But if you remember, we said, okay, the process, the population size in this case must be correlated over time, but the observation process shouldn't. And if we drew a line straight through the data, then we would have possibly a bunch of points under the line, and then we would have a bunch of points above the line, and then we would have a bunch of points under the line. And that would mean that our observation error is actually autocorrelated. So the model says, no, okay, we have described the process being autocorrelated, so it must be the process that's weakly and not the observations. Another interesting feature that we see here is that, for example, between 2010 and 2015 or something like that, we didn't have any counts. And so we can see that the model can handle that, but the uncertainty grows, obviously, in this period where there's no count. And then we can also go to the lower panel. Oops, sorry, let's press this. We can go to the lower panels, and this shows you how the population is evolving. So this would be, if we had a 0 line here, that would mean that the population is not changing. But whenever the line goes above 0, that means that the population is growing at a certain rate in that period. And here, this would be the proportion of birds that are present in the winter in relation to the summer. So that would be, if we have a negative number here, that means that our species is more abundant during the summer months. And if we would have a positive number over here, that would mean that our species is more abundant in the winter months. So this is a preliminary result of what we should expect. And then I'm going to hand it over to Res, that is going to explain a little bit more the occupancy ones. Res, go ahead.
Thank you, Pachi. Yeah, so I'm going to speak about how we analyze the SABAP data using occupancy models. So with the SABAP data, we are mostly interested in whether a species occurs in the site or not. And with occupancy models, we recognize again that we have the hidden sort of true state of the system that we are interested in. And it's that in the left-hand diagram, that set plane there with the bunnies that you can see. But what we actually observe is a little bit different, because in certain places we might go look for the species and then not see it. So we basically overlook it. And so that observation process again is important, and we are using models to try to estimate that observation process and account for it. So we need some information on the observation process, and in the case of the SABAP data, that comes from repeated observations at the same sites. Next slide, please, Pachi. So this is just to quickly remind you about the SABAP data. So the SABAP data are collected at a 5x5 minute grid across the entire country. So we are focusing on the water birds here, but these methods work on all the species really, as long as there's some decent data. Next slide, please. Okay, so the next few slides, I'm going to quickly explain how occupancy models work. So in the diagram, the top left block represents a grid, an area, and the species occurs in those grid cells that are colored in orange. So this is the true state. This is what we want to know. But now imagine we go to these places and we just record whether we see the species or not, and what we get is the other three parts of this diagram. So for example, on the top right hand side, that will be the first visit to each site, and you can see at some of the places we detect the species in black, and then others we don't detect it. And then when we go again, we might see it again in some of the places that we saw it before, in others we might not find it now, and so on. So like, for example, if you, yeah, so so the main point that you can see immediately here, the main assumption is that the true occupancy status stays the same while you're sampling, so that the top left graph is the truth and it doesn't change. And that way, because we repeatedly sample, we can then gain some information on the observation process. The other key assumption that you can see from this slide is that we are not allowing for any false detections. So if someone reports a species from a site, we assume that that's correctly identified. And that's really where the vetting procedure becomes really important. So that the no false positive assumptions could be relaxed. And the closure assumption, which is the other one I mentioned earlier, can also be relaxed. But in a basic occupancy model, we make these two assumptions. Next slide, please. Yeah, so this is really just showing you how we set up the model, and it's a type of state space model. Just that instead of having time now, we have space. So we've got that underlying true occupancy, which is which we denote with Z. So it's either a site is occupied or not, and that follows a Bernoulli distribution with probability psi. But that's not what we observe directly. What we see are the Y's. So we either detect or don't detect the species at a given site during a particular survey. And that again follows a Bernoulli distribution, but now dependent on the true occupancy status. So if the species is there, we detect it with probability P. If it's not there, we don't detect it. Next slide, please. Now, the important thing is, so occupancy really is the proportion of sites that are truly occupied. But what we are interested in is how this varies across space. So we want to know which sites are likely to have the species and which ones don't. And so it's quite important for us that we have informative covariates. And the purpose of this slide is really just to say that we have two types of covariates. We've got the covariates on occupancy, and then we can also model variation in the observation process. So variation in that detection probability P. Next slide, please. And here are just some quick examples of what these covariates could be. So we've used these models quite a bit on the SABAP data. And so for example, for occupancy, we might use climatic data like growing degrees, minimum temperature, and then some measures of dryness. For detection, the observation process, we have other type of information, like for example, the number of hours spent birding, which season it was, things that we think affect the observation process particularly. And Jim Nichols and I recently published a review paper on these methods used on citizen science data in methods in ecology and evolution. Next slide, please. So I mentioned that closure assumption, that's really important, but obviously what we want to know is not just where the species occurs at a given time, but also how occupancy changes over time. So what we are really interested in is, are species disappearing from certain areas, or are they colonizing new areas? And so we can relax that closure assumption by assuming closure only over shorter time periods. So for example, a year or a few months. And then between those particular periods, we allow for local extinction and colonization. So these are the dynamic occupancy models. And it's quite exciting. So we can look at, we can estimate extinction and colonization probabilities and look at drivers, covariates of those as well. Next slide, please. And here's an example where we use these models to look at the range expansion of the common myna. So common myna is an invasive species, and it has been in South Africa for quite a while, but you can see from the SABAP data there on the right-hand side. So top left is 2008 and top right 2009 and so on. And you can see how the species is still expanding quite rapidly from year to year. So it's expanding into the northwest and also Kruger National Park. And so we can use these models to to get a quite detailed description of the spatial range dynamics of species. Next slide, please. Yeah, so that was an explanation of occupancy models. And so I just want to shift gears a little bit. So Nancy initially spoke about how we engaged with the stakeholders to try to understand what kind of decisions people need to make. So what we want with this pipeline is really to provide something that's useful for people who are supports them doing things that they are already doing, not something extra. And what came out quite strongly is that they need trends and they need occupancy status, and then and trends in occupancy and trends in abundance. So basically, those are the main things. And so we are thinking, and then the other important thing that came out is that those BIRD variables need to be scalable across space and time. So some decisions are made at a short, sort of frequent time frame, others at a more at longer time frames, maybe once a year, once every 5 years, or even less frequently. And then also across space. So a lot of managers said, for example, they see a trend at their local wetland and they want to see whether that trend actually mirrors something, a regional trend, something that happens at nearby wetlands as well. So we need to be able to scale across space. Okay, so one of the things that we can calculate from these occupancy probabilities is species richness. So that could be just a sum of the occupancy probabilities. But we are also looking into more sort of, yeah, maybe numbers that are more informative for other types of things. And the Hill numbers are a really flexible tool here. So the Hill numbers is, yeah, that formula there looks maybe a little bit intimidating, but it's a diversity measure that includes, so for example, it has that Q parameter. If you set that Q to 0, then it returns something that's equivalent to species richness. But then there are also these other diversity measures that also account for variability in abundance of the different species or occupancy probability. And if you set that Q to 1, so we need to take the limit there because otherwise we get an exponent of 0, where we have something that's equivalent to the Shannon diversity. And then if we set that Q to 2, it's equivalent to the Simpson diversity. So it's just different ways of weighting components of species richness versus evenness, basically. Next slide, please. Yeah, and then we are looking into ways of aggregating trends across species and sites. So for example, you might be interested in all like trends in all the long distance migrants at a particular site, or so then you need to be able to combine the trends across the species that are long distance migrants. Or you might be interested in a trend of a particular species, but at multiple wetlands, and then you want to aggregate the trends across different wetlands. And so we've started looking into things like, for example, the Living Planet Index really just uses the geometric mean. So that's a mean of the log growth rates or log trends. So that would be really very easy to get out of the state space models that Pachi presented. They could be weighted in different ways. So the Living Planet Index itself is kind of starting at an arbitrary place somewhere not that they define as 1. We're not necessarily thinking about that. We're more maybe thinking about something that's maybe more akin to the stock market index. So what we want is really an index that tells people, the water birds are going up or down, and then people can drill down and can say, well, this trend is actually driven by maybe the long distance migrants. So that might indicate if it's all the long distance migrants, maybe something wrong at a different place, not my wetland, or it might be driven by counts that come from dams, or it might be driven by counts that come from particular types of wetlands. So we want the index to be sort of give a quick overview, but then also be able to disaggregate and actually see what the causes are of those changes. Yeah, next slide, please. And then another important thing is how we communicate uncertainty. So all these estimates and measures come with a degree of uncertainty, as you've seen now, and how we actually communicate that in a way that makes doesn't complicate the message basically, but makes it realistic. So if you see a trend, is this a real trend, or is it maybe just noise? And if you don't see a trend, can you be sure that in reality nothing has happened? Next slide, please. Yeah, and that really brings me to the end of our presentation. So we are working on that sort of middle section of the data pipeline, where we want to add statistical tools that basically draw information out of the raw data. So we want to enrich that pipeline with not just raw data, but indicators or indices that give management relevant information. Thank you.
Thanks very much, Rez.